Hi everyone!
You're listening to SciPub,
where we are talking science.
Not only for the experts, but for everyone.
Welcome to a new episode.
Hello everyone, this is Diana and Sergi and we are presenting SciPub with our guest today, Joshua Frenster, who is a postdoctoral researcher in Stembryo engineering group at UPF, and he will be here to answer our questions and talk about mainly stem cells and early development. So let's get started.
Thank you so much for being here, Joshua.
Yeah, thanks for inviting me. Very cool project.
Thank you. So we will start with some general questions about stem cells, early development. So if you can tell us what stem cells are, what are embryonic stem cells, because I know that you're working with
Very
these.
nice.
And maybe what is the difference between them, and what is the difference between stem cells in general and our normal cells?
Yeah, cool, great intro question. I think it's great that you started differentiating embryonic stem cells and stem cells because there's different types of them. Maybe if we start with the normal cells, those are the differentiated cells in our body, the cells that specialize into a certain function. So they change their inner programs and their shape and their function to do one specific job. Right, the skin cells are your protective layer, your intestinal cells absorb nutrients, your neurons fire action potentials, but they basically kind of specify into one thing. Stem cells are cells that still divide and grow, and they have the potential to become one of those differentiated cells, but they have not really committed to anything yet. So if we start with the embryonic stem cells, those are the cells in early development that are basically the start of it all, right? You start with one single cell that grows into a couple of cells. Those pluripotent cells are your embryonic stem cells. Pluripotent because they have the potential to become many different types. Actually, in fact, all types in your body. But again, they have not committed yet. And the adult stem cells are those that can still make a few types. They would usually be multipotent or unipotent to make one specific type, but they can still self-renew. They're kind of your reservoir to replenish a tissue.
Wow, nice. Thank you. And would you say that it's possible to turn a normal cell, again, a differentiated cell as you said, back to its pluripotent state, so back to a stem cell?
Yes, absolutely. And I think that's really a cool and fascinating thing that's somewhat recent. There was actually a Nobel Prize in that in 2012. So that's a process called reprogramming, where you try to basically revert the changes that specialize the cell and open it up again to regain the potential to do all types of the cells. So the most common way of doing that would be by overexpressing certain transcription factors, so changing the gene expression patterns of a cell to mimic what happens in a pluripotent cell. And if that happens long enough, some of the cells actually kind of adapt to that state and become pluripotent again. Still very inefficient. It's 1 in a few thousand or 1 in a million or something like this in the laboratory, but it is possible in principle, which is really cool. So yeah, it is possible. It's not as efficient as it could be, but it's possible.
It sounds really cool. And how about the use of stem cells in therapy and in research? Can you tell us a bit more about it?
I think the use of stem cells in research is really important because it helps us to understand how these cells make the decisions to become differentiated cells. So if we think about a certain tissue, right, if you have like some stem cell pool, for example, in the skin, you have these stem cells that self-renew, multiply, and then every once in a while differentiate to make skin cells. But this process of how the cell decides to self-renew itself and just divide, or to make a specialized cell, how different stem cells make these decisions, and especially in development, how the embryonic stem cells basically shape our entire body. I think that's why we use them in research. And especially because we can't study this in the womb, we can't study this with whole animals as easily, we try to use stem cells to understand this decision making in a dish, but also to understand generally how our bodies are built, basically. For therapy, that's not exactly my expertise, but if we keep in mind that stem cells specifically are able to grow and self-renew, but also make specific cell types. You could say that the great thing about a stem cell is that you could bring it to some place, it could grow, make more, and then create those functional specialized cells. So if you now have a disease that is, for example, caused by a mutation that could be easily repaired, so a great example is sickle cell anemia in the blood, where a single mutation in the stem cells that make up the blood make the specialized differentiated cells defective. So one idea is that if you could take a stem cell of the patient, fix that one mutation, and bring that stem cell back into the patient, you could have a new stem cell now that replenishes and makes differentiated cells again, but without the defect. So basically it's a way to repair or reset a certain lineage in your body that may give rise to faulty cells and replace that with cells that now give rise to repaired cells. But caveat is, I know there's a lot of popular science about injecting stem cells and fixing everything.
Yeah.
Injecting stem cells into random places is not going to be a solution for anything. So I think there's a few examples where it works really well and many examples where it's not going to help in any way.
Or it can even make things worse from time to time. No?
If you do it wrong, for sure. I can imagine.
And one last general question. Can you tell us a bit about early development? I know that's part of your interest, so if you can explain what it is and maybe how it goes.
So early development, so early embryonic development is really the process of how a body is, well, created is the wrong word, but the process of how a body is shaped. So if you imagine you start with a single cell, a fertilized egg, that divides, it makes a couple of cells, now you have this few hundred cells, which are the embryonic stem cells. Those are the pluripotent cells that can create the entire body, but have not decided to do anything yet. Then there's a process called gastrulation, in which these cells now start to stratify, make the three main germ layers of the body, so neuroderm, neuroectoderm, let's call it ectoderm, ectoderm, mesoderm, and endoderm. And how they then continue to create the body axes, right? So this, let's call it a bunch or clump of cells, has to decide what's going to be front, what's going to be back, what's going to be up and right, who's going to make what, and how do they know what the others are making and coordinate what they make in response. So I think it's a really complicated process that really explains how, from a single point, you create this complex body. And I think fascinatingly, in most cases, it goes really well, and the bodies look somewhat similar in rough shapes, right? Few of us are spirals. And that's why I think it's a really fascinating process that I think we still understand by far too little about.
For sure, thank you.
So, getting more into your research specifically, so we know you are using gastroloids in your research. Can you explain us a little bit what are these and what do you use it for, like generally first?
Gastroloids are a stem cell based in vitro system that we use to model exactly this process of gastrulation. So in the embryo, usually you have these aggregates of embryonic stem cells that then together make the decision to start breaking the symmetry. In the beginning, they're all the same, they're kind of a bunch. And then they say, okay, I'll make the front, everybody else respond to that, I'll make the back, I'll make up, down. The cool thing about gastroloids is that they undergo the same developmental processes as an embryo would, but without actually forming an embryo. So we start by using stem cells, we aggregate them in a dish and give them certain growth conditions, which allows them to kind of self-organize, undergo the same motions, they make the three germ layers again, they create a body axis. So actually in the dish you'll see there are aggregates of cells that grow and at some point they elongate, a little bit like a little bean that makes a body. But without creating an entire body, it's a part of an embryo. And it helps us study the decision-making of the cells during this process, the coordination of the cells, of basically breaking the symmetry, becoming not the same but different, and talking to each other in the process.
It's very cool. And if I can ask, how much can you recapitulate, like until which stage?
Depends a little bit on in which direction you try to go. So in our lab we use gastroloids, or in my case mouse gastroloids. You can bring them to roughly what in the mouse would be the counterpart of a day 8 or 9 embryo. If you go into specific lineages, so if you try to bring them into one specific tissue, say you want to create cells of the heart or cells of a gut, then we can go a little bit more further in specialized ways. But one important thing to keep in mind is that all of this is not an entire embryo. It's always just a part. In our cases, in the classic gastroloids, we don't get head structures. So basically it mimics these early processes, what happens in the mouse around day 6. In the human maybe weeks 2 and 3, those processes we can model, but without creating an embryo.
That's really cool. And getting also more in what you do, we know that you are investigating competition, cellular competition in the development. So can you tell us a little bit about what this is and what are you doing?
Developmental cell competition is a quality control step in early development. So at a similar time point that I mentioned earlier around gastrulation, when you still have a group of cells that is pluripotent and to some degree all the same, just before they decide splitting up into the different parts of the body, they compare their relative fitness to each other, meaning they perceive to some degree how healthy or how intact are they. They communicate to the neighbors how intact are the neighbors, and then depending on that they may decide to continue forming a body, or they may undergo suicide to make space for other better cells. So it's kind of a process that is like a quality control where you can still get rid of bad cells, or rather where the bad cells get rid of themselves. And it's a process that we know very little about. So it's happening in early development, in the mouse around day 5, in the human probably around, I don't even know. That would be maybe around 2 weeks of embryo. And I think it might be one of the steps that gets rid of a lot of faulty cells that you have in early development. So basically, in the first couple of days, a lot of errors happen during development, especially spindle issues and segregation of chromosomes. So you get a lot of aneuploidies, cells that have too many or too few chromosomes. And I think that this is one of the main steps where you can still easily get rid of those cells before you start specifying them into a body. So as long as you're all the same, it's easy to get rid of a couple. If you already decide who does what, it's very difficult to lose something because suddenly you lose a part of your body.
And do you know how much about this signaling between these cells we know, or if we can know more?
Yeah, there's astonishingly little known about this process. So overall, cells talk to each other all the time. Cells are constantly shouting out and listening. There's different ways of signaling. They can signal via secreted molecules far range, so they can basically send out messages that get received all around them. Or they can have contact-mediated communication, so little proteins on the surface of a cell that touches the neighbor cells. This way they communicate. So generally they're communicating all the time, but in cell competition specifically, we don't really know how they communicate, at least not in mammals. So we do know certain things about how they measure fitness, or let's say how fitness is perceived or defined, but we don't really know how they compare that to the neighbors. And I think that kind of gets to the core of what I'll try to find out is, as a cell that is, say, somewhat okay, how do you know whether or not to kill yourself in response to your neighbors? So I think that's something that we don't know. A lot of the research was actually done in fruit flies. So the cell competition field is kind of a 20, 30 years old or something from the fruit fly, but only very recently has moved into mammals and recently mice. So I think we're just still starting out studying it. We're just realizing how big of a field this is.
Yeah, of course.
Well, one thing that I didn't mention, cell competition kills up to 30% of the embryo. So it's a very drastic, I think that's something that a lot of people don't know, is that basically in this early, say, 2-week stage of a human, up to 30% of the embryo might undergo apoptosis, kill themselves to make space. And people don't really understand how that happens. So I think important to learn a lot more about it.
Yeah, exactly. And if it's like such a big process, of course. So just one last specific question. Can you tell us something more about your own research, or what are you working on? How is your project going, or something like that?
Yeah. So what I specifically study is cell competition in early development, and I use gastruloids to study that process. So my question is, how do the cells communicate to each other, figure out how fit they are themselves, how fit their neighbors are, and whether or not they should undergo suicide to make space for their neighbors. I use gastruloids, this in vitro model system, which is cell-based. It's basically a part of an embryo undergoing development in a dish, which means that I can watch this process of how the cells undergo the developmental processes, and then how some of the cells may die or may not die in different conditions. So basically I try to understand how the cells make the decision to die, and ultimately how the cells communicate to each other to decide who should die. And I use gastroids for that because it's a system where I can observe the cells, I can let them develop and grow on the microscope, so I can observe live what's happening. I can put different fluorescent colors in it, so I can watch, say, cells that I want to die versus cells that shouldn't die in different colors and see how they interact. So it's really, I think that the gastroid is a system that helps me study the process while being able to observe it and to modify it.
It's very nice.
Yeah. And what would you say it's like the importance of this research for, I don't know, the world and
for the world? I think the importance of this research is, first of all, that we understand the development process that I think plays a very important role and has been neglected a lot. It's very early on in development. What many people don't know is that I think up to 30% of all conceptions are actually lost in the first 2 to 3 weeks. So long before a person would know that they are pregnant, they may already have lost an embryo.
Mm-hmm.
So I think there's a lot that happens very early in development that people are kind of oblivious to. And I'm trying to understand a quality control step in this moment to see how the cells find issues, fix issues, and how development can undergo normally. So I guess this touches upon loss of birth, birth defects, and overall health of development.
Awesome, thank you. And yeah, we went to the streets and we asked people what do they want to know about this topic. They were mainly focused on stem cells, I think, probably because it's something that is well known to them, or at least a little bit, they know what it is. So we had some questions. First question is, do you think that in the future stem cells use will be more present than it is today? So will we use stem cells more in future, basically?
Absolutely. I think the stem cell field overall, I mean, it's not a young field anymore, but I think it's still a field that is growing very fast and still has a lot ahead of itself. I think technically there's a lot of new things that we're able to do now that we study. For example, the gastroids, that's a kind of new thing. The way that we can mimic development of an embryo in a dish without making a whole embryo. And study these processes outside of an animal in an ethically clean way, I think enables us to do a lot of more studies that we couldn't do before. Therapeutically, I think the more we understand the processes, the more we understand the cells and learn how to work with the cells, the more we'll be able to do. And overall, I think it's going to be a field that keeps growing, hopefully. And I hope in the order that we do the research first and then the therapy and not the other way around, to do things right.
Nice. And one person wanted to know, is it possible to use stem cells in cancer therapy? Is that a thing?
That's again not exactly my expertise, but so one way that you could leverage stem cells for in cancer therapies would be, again, replacement. So similar to what I mentioned earlier is if you have, say, cells of a certain cell type or lineage, where the stem cell of that lineage has a certain mutation or defect. One thing that you could do, if that mutation results in cancerous cells, one thing that you could try to do is basically get rid of that lineage in a patient and reintroduce either repaired or healthy stem cells to replace that lineage. So that's, I think, especially a thing in blood cancers, where if you have defects, the stem cells keep producing more defective cells. If you could try get rid of all of those cells, you get rid of the source of the cells in a way, and reintroduce healthy stem cells, then those healthy cells can replenish the blood cell line or cell lineages.
And another person in the streets was concerned about non-controlled proliferation of stem cells, especially when you use them in therapy. Can you tell us more about it?
I think that the worries of that comes from the fact that both stem cells and cancer cells have in common that they can grow, right? So stem cells self-renew, they can divide and become more of itself in a very controlled manner. In cancer cells, the issue is that they can also divide and make more of itself in a less controlled manner, and that is where the disease comes from. That's why I think it makes sense to be worried about this. If you were to inject stem cells into random parts of the body, in most cases your immune system would kill them, the cells would differentiate and form something, but probably not grow very much. But I think keeping in mind that a stem cell is able to grow and proliferate and make more of itself is something to keep in mind, and that's why injecting stem cells into random regions It's not going to be a general therapy.
So now to end a little bit this, I want to ask you some questions about your personal career. So as I said, you are a postdoc at UPF, and can you tell us about your background or how did you get here?
Yeah, so my background is I've done my Bachelor's of Science in Germany. So originally I'm German. I've lived there until the Bachelor's and then moved to New York, where at NYU I did my Master's and PhD. The topics there were mostly cancer biology, so I worked with a neurosurgeon in trying to study and understand glioblastoma, so very aggressive brain cancers, and how we could find new therapies against those cancers. But also very specialized on receptor biology, so trying to understand how those cells communicate. So there's a couple of themes that are common with what we do now. In this cancer field, I also focused on cancer stem cells, which are a somewhat controversial topic. That some of the cells within a cancer are also creating lineages, are self-renewing more than others. And I think that's why a lot of the courses I took during my master's were focusing on stem cells, rotations, and reprogramming labs. So I think the theme of stem cells has been around for me for a very long time. Then I focused on cancer for a very long time and receptor biology. Now I think I'm coming back to very pure stem cell biology. But at the same time, I realized that in the topic that I work on now, there's a lot of commonalities with the cancer biology. So the way that the cells in cell competition perceive their fitness involve genes that are classic tumor genes, classic oncogenes or tumor suppressors. The way that they communicate to each other goes a lot into the cell-cell signaling studies that I've done before. So I think in a way, the postdoc that I do now combines a lot of the topics that I touched upon before and brings them into a different context. Yeah, and I've been here as a postdoc for 2 years now.
Okay, that's very good that you can have like background in different areas, but you realize that at the end everything is useful to your.
I think in the end, I mean, what I study is cells, and cells do different things.
Exactly.
And once you learn something about the cell, you apply it to a different field. It's still going to be the same actors in your game, right? The cells do cell things.
Exactly. So after your postdoc, for example, what are your further plans, or what do you want to focus more on?
The plans for me are going to be to hopefully become a group leader. We'll see how things go, but my plan would be that after the postdoc, I apply for faculty positions, start my own research lab. I still like this overlap of, say, developmental biology and cancer biology. I don't know where that's going to be, but I'll hopefully find out in the next couple of years.
Okay, so now to end this podcast, I wanted to ask you to give some advice to the public and also for people who is starting their own career, so students. What would you say to them, or what's your advice for their scientific career?
All right, for the public, trust the researchers who do the actual research. I know that there's a lot of misinformation. A lot of people say studies have shown that. If they don't tell you what study that is, don't believe them. Especially, I think, in the stem cell field. It's very quick that bad news make headlines, things get misinterpreted. Read commentaries and things that actual scientists wrote and not people wrote about scientists. I think that's going to be very important. Yeah. For students, I think for young researchers and students, one of the most important things is find a mentor that wants to support you in your growth. Find a mentor that will help you make your pathway in science. Not somebody that you think is great and you'll get attached to them, but somebody who thinks you could be great and who will make you great. And do the research that you love. Science is a tough job. There's few benefits to it, but if you do the research you love, then that's going to be your reward.
Thank you very much. This was very wise advice.
Thank you.
And thank you so much for being our guest. It was really nice talking to you.
Yeah, cool. Thanks for inviting me. Super fun.
Yeah, we hope that our listeners have their doubts solved, what they ask in the streets and similar things. And I totally learned something new.
Me too. A lot of interesting facts that I have never even thought about.
Yeah.
Cool. That's perfect.
Yeah, so I think that's it.
Yeah,
Thank you so much for being here, and see you.
Cool.